You know, for decades, whenever we talked about aging, there was this universal, almost comforting analogy we all just sort of defaulted to.
Oh, right, the rusty car.
Exactly,
Right.
rusty car. You drive it long enough, and, you know, eventually the brake pads wear down, the transmission starts slipping.
Metal oxidizes.
Right, and when your knees creak in the morning or your eyesight dims, it feels exactly like that. It just feels like physical wear and tear.
Yeah, entropic decay.
Yeah. You assume it's inevitable because physical parts just, well, they break down over time. But welcome to today's deep dive, because we are looking at a stack of research, primarily this massive, honestly mind-bending paper called Biochemical Architectures of Systemic Rejuvenation. And it throws that entire rusty car metaphor completely out the window.
It really does. It requires a complete rewiring of how we view our own biology.
Right, because aging, according to this new paradigm, is not a random loss of physical parts. It is a programmed, and more importantly, a reversible loss of information.
Exactly. And to understand how profound this shift is, we really have to look at the epigenetic theory of aging.
Okay, let's unpack this. What does that actually mean for the listener?
So think of your DNA, your genome, as the physical hardware of a computer.
Okay, like the circuits.
Right, the physical circuits, the microchips, the hard drive. But that hardware is absolutely useless without software.
Right.
Your epigenome is that software. It's the operating system that tells your cells exactly what to do, which genes to turn on, which to turn off, and crucially, how to repair themselves.
So the overarching mission of our deep dive today is to explore how this biological software gets corrupted over time.
Right.
And to look at the incredibly precise toolkit scientists are building to essentially debug and rewrite it.
That's it, exactly.
So we aren't talking about the DNA mutating or like physically breaking apart.
No, the hardware is fine.
We are talking about the cell just forgetting how to read its own instructions.
Precisely. And the paper introduces a concept to explain this called DMI, or DNA methylation instability.
And there is a hyper-specific number in the research here that is just, it's fascinating to me.
Oh, yes. The 31,744 stable methylation sites.
Yes, that exact number.
Yeah. In healthy, youthful individuals, researchers have identified exactly 31,744 of these specific locations on your DNA that they must remain unmethylated.
Meaning they need to stay completely open.
Open, transparent, accessible. You can think of them as sentinel sites. As long as they are clear, your cells can easily read the instruction manuals for maintaining cellular identity and executing, routine repairs.
Right, but biological weathering, which is the actual physical sensation of aging that you, the listener, experience, is essentially the hypermethylation of these exact sites.
Exactly. The body starts placing chemical blockades, these methyl groups, right over the sentinel sites.
Okay, sticking with the software analogy, this makes perfect sense. It's like your computer's operating system slowly, over the years, placing encrypted passwords on your most critical system repair files.
That is a perfect way to visualize it.
You click on the Fix It folder and it's just locked. The hardware underneath is completely fine. The instructions are still there.
But the software absolutely refuses to let the cell access them.
Right. And the paper gives a really tangible example of this with a gene called ELO-VL2.
Yeah, ELO-VL2 it's the perfect illustration of how this software corruption translates into physical decay. This specific gene is actually such a reliable indicator of this process that it's used as a highly accurate predictor of a person's chronological age.
Wow.
Yeah. When the ELO-VL2 gene gets locked down by those chemical blockades, the body loses its ability to synthesize very long-chain polyunsaturated fatty acids.
Which sounds like a mouthful, but we are basically talking about DHA, right?
Specifically DHA, yes, which is a structural component absolutely vital for tissue health, particularly in your retina.
And this is where the science stops being abstract and becomes incredibly real for you listening.
Yeah.
Because this isn't just a theoretical model of aging.
No, not at all.
This is the exact mechanical reason why your vision degrades as you get older. The cell can't read the ELO-VL2 file, so it stops making DHA, so your eye tissue physically starts to fail.
But here is the most important part of that experiment. Researchers wanted to know if the physical damage was permanent.
Right.
So they bypassed the locked ELO-VL2 gene completely. They simply took the product it was supposed to make, the DHA, and directly injected it.
And what happened?
By doing that, they actually reversed the retinal aging features in the experimental models. They restored visual function.
So by just providing the missing output, the hardware started working perfectly again.
Exactly. Fix the software output or clear the blockades and the biological machine runs youthfully again.
It proves the rusty car theory wrong right there. The eye wasn't physically broken beyond repair. It was just starved of its required software output.
Right.
But, this leads me to a darker question about the cellular environment. If the software is locking down these 31,744 sentinel sites, what actually happens to the cell itself?
That's the million-dollar question.
Like, if a cell is completely blinded to its own repair manuals and loses its identity, does it just quietly die off and make room for a new healthy cell?
You would hope so. I mean, the clean, programmed death of a cell is called apoptosis, and it's a very healthy, normal function.
Right.
But when the software corrupts, particularly in genetic regions controlled by something called the PRC2 axis, that clean disposal doesn't happen.
Okay, what is a PRC2 axis?
So the PRC2 axis normally regulates the genes that tell a cell what kind of cell it is, whether it's a skin cell, a liver cell, or a neuron.
Oh, it's identity.
Exactly. When those identity genes are abnormally silenced by the software corruption, the cell experiences massive stress. It essentially forgets what it is.
Wow.
And instead of dying, it enters a state of permanent arrest, a senescent state. It becomes a zombie cell.
And a zombie cell isn't just inert. It doesn't just sit there taking up space. It actively causes harm, right?
Oh, definitely.
It secretes this toxic sludge into the surrounding tissue.
In biology, we call it the SSP, the senescence-associated secretory phenotype. But, frankly, toxic sludge is a very accurate way to visualize it.
Good to know my terminology holds up.
It really does. It is a localized, highly inflammatory chemical storm. These zombie cells are essentially panicking, and they release inflammatory markers that damage the healthy neighboring cells.
So they're effectively accelerating the aging process of the entire tissue around them.
Exactly.
So dealing with these zombie cells seems like priority number one. And the paper details two very different pharmacological approaches to this.
Right, the xenomorphic and senioriverse approaches.
Let's start with the first one, the xenomorphic approach, which uses something called the E5 cocktail. From my understanding, this cocktail is essentially a highly targeted sedative.
That's a great way to put it.
It uses specific microRNAs to override the internal panic alarms of the cell. It suppresses the genes responsible for triggering that heavy inflammation.
Yeah, the E5 cocktail doesn't kill the zombie cell, but it neutralizes the threat. By heavily suppressing those downstream inflammatory responses, it stops the cell from secreting the toxic SSA sludge.
So it puts the zombies to sleep.
Right, which immediately lowers the systemic inflammation in the tissue and extends the health span of the organism.
Okay.
But there is a second, far more radical approach outlined in the research called the senioriverse strategy.
Okay.
This uses a specific molecule called miR-302B, which is derived from embryonic stem cells.
Hold on. Let me stop you there. Because the senioriverse strategy tries to actually rehabilitate the cell, right?
Yes.
If these zombie cells have been sitting around secreting toxic venom and destroying their neighbors, my instinct is that we should just find a way to destroy them completely.
Just wipe them out.
Yeah. Flush them out of the system. Why on earth would we risk waking them back up and rehabilitating them?
It's a very logical instinct. The problem with simply destroying all senescent cells is that as you age, you have a lot of them.
Right. That makes sense.
If you aggressively kill them all off, you risk depleting your tissue mass and exhausting your stem cell reserves because your body has to work overtime to replace all the cells you just destroyed.
Oh, wow. I didn't think about that.
Yeah, so the senioriverse strategy asks, what if we could just fix their software instead?
Okay, so how does miR-302B actually do that? How do you cure a zombie cell?
It targets the epigenetic erasers. These are specific proteins that are actively holding the cell in that arrested zombie state.
Okay.
The miR-302B molecule essentially inhibits these erasers. By removing the proteins that are enforcing the lockdown, the cell's natural cycle inhibitors are suppressed.
So the cell just wakes up.
It wakes up, realizes it has access to its instruction manuals again, re-enters the normal cell cycle, and restores its youthful ability to divide and function. It's true cellular rehabilitation.
Which is an incredible concept, but, it brings us to a huge logistical problem.
What's that?
Even if we use this senioriverse strategy to wake these cells up and rehabilitate them, they are still floating in a completely degraded neighborhood.
Oh. Yes.
They've been pumping out that toxic SSP sludge for years. Before we can trust these newly functioning cells to do their jobs, we have to clean up the environment. We have to clear the static.
You're exactly right.
And this brings us to a fascinating part of the research, because this isn't just happening in petri dishes or mouse models. This is a current clinical procedure.
Yes, the system-wide flush. To clean the environment, the research highlights a landmark clinical trial out of the Buck Institute, led by researcher David Furman.
Right.
They used an existing clinical procedure called therapeutic plasma exchange combined with intravenous immunoglobulin. We usually abbreviate it to TPE-IVB.
For you listening, picture this practically. This involves physically hooking up to a machine that separates your blood plasma from your blood cell.
Exactly.
It filters out the plasma, which contains all those circulating inflammatory markers, the gerontogens, the toxic sludge the zombie cells left behind, and replaces it with fresh, clean fluid.
And the data from doing this is remarkable. The trial showed that a biweekly TPC-IV regimen reduced the biological age of the participants by an average of 2.61 years.
2.61 years. That is insane.
And they didn't just guess that number. They measured it across 15 different highly calibrated epigenetic clocks.
Almost 3 biological years reversed, purely from taking the trash out of the bloodstream.
Because when you remove that heavy, suppressive, toxic sludge, the body's natural systems rebound immediately. The researchers described it as a coordinated regreening of the immune system.
Regreening, I like that.
Yeah. When they looked at the blood profiles of these older patients after the plasma exchange, their proportions of naive T-cells and natural killer cells have shifted. They looked remarkably like the immune profiles of much younger people.
But logically, this systemic flush must have different effects depending on who is receiving it. I mean, does a relatively healthy 30-year-old get the same benefit as someone much older?
This raises an important question, and the clinical data is very clear on this. The most profound rejuvenation responses were seen in individuals who had the highest baseline of biological weathering.
Meaning they were in worse shape to begin with.
Exactly. We are talking about people who started the trial with high circulating levels of bilirubin, elevated glucose, high liver enzymes, people whose systems were under massive metabolic stress.
Right.
For them, TPE acts as a profound systemic cleansing mechanism. By clearing the sheer volume of inflammatory static, it allows their own native adaptive repair capacities to finally kick back online.
Okay, so let's track where we are. We figured out how to patch the corrupted software.
Yep.
We've rehabilitated the zombie cells back into the workforce. We've completely filtered the toxic sludge out of the plasma, which has naturally regreened the immune system.
Exactly.
Here's where it gets really interesting. Because once those youthful immune cells are back online and circulating in clean plasma, what do they actually do?
Right, they don't just fight off colds.
No, they build an incredible internal delivery network.
They do, and it might be one of the most elegant discoveries in modern biology. They call them telomere rivers.
Telomere rivers.
Yeah. We used to think immune cells just circulated passively, waiting for an infection. But groundbreaking research shows that rejuvenated CD4 plus T-cells actually coordinate a systemic youth program.
Wow.
They physically create these tunneling vessel-like networks that connect to senescent tissues across multiple different organs. And through these rivers, they distribute critical supplies.
I was trying to picture how to explain this to the listener, and at first I thought about a package delivery network, like Amazon Prime for your cells.
That's not bad.
But really, it's more like a highly advanced biological irrigation system. You have these rejuvenated immune cells acting as localized water towers, and they are pumping vital nutrients, things like telomeric material and stemness factors, directly to the dying crops, the aging tissues, that desperately need them.
The fertilizer in your irrigation analogy would be crucial youthful proteins like WNT and beta-catenin. The T-cells package these up and send them down the rivers. But just like any physical irrigation system, it requires massive amounts of energy to run the pumps.
Right? Where does the power come from?
The engine driving these telomere rivers is a process called fatty acid oxidation, and it relies heavily on a very specific rate-limiting enzyme known as CPT1A.
CPP1A.
Right. For the T-cell to sort and package these youth factors correctly, ensuring it only sends the good stuff and excludes metabolic waste, it requires absolute energetic precision from that CPT1A enzyme.
And as we age, that engine starts to fail. The CPT1A enzyme activity declines.
Exactly.
And because the T cells can't burn fat for fuel effectively anymore, they start hoarding it. They accumulate this specific type of fat called ceramide.
Yes, ceramide.
And then the pumps get completely clogged, the irrigation system breaks down, and the telomere rivers dry up.
Precisely. The youth factors never reach the aging organs. But researchers asked, what if we just fix the pump?
How do you do that?
They used CPT1A activators drugs like fenofibrate or a class of molecules called DOS disruptors.
Okay.
By administering these to aged mice, they cleared the ceramide clogs. They restored the enzyme's ability to burn fat. The pumps turned back on, the rivers flowed again, and the localized tissue repair resumed.
And the result of turning those rivers back on, they extended the median lifespan of those mice by 17 months.
Yeah, 17 months in a mouse model is a staggering health span extension.
It's massive.
Yeah.
But, it brings us to a critical pivot point in this deep dive. We have the rivers flowing. We have the software patch. The plasma is clean. How do we protect this beautifully complex system from just decaying again tomorrow?
Right, you have to maintain it.
Yeah, how do we build a biological shield?
The paper outlines a fascinating dual approach to maintenance. You need a broad metabolic shield, and you need precision hardware resets.
Okay.
For the broad shield, the research points to GLP-1 receptor agonists.
Which is a term you listening have almost certainly heard in the news recently regarding blockbuster weight loss drugs.
Oh, absolutely.
But their role in longevity and systemic rejuvenation is completely different.
Entirely different. In the context of aging, GLP-1 arrays act as a metabolic shield. They dampen the systemic inflammatory noise and actively protect the epigenome from that random software corruption we talked about earlier.
How do they do that?
They do this by activating the AMPK pathway, which is essentially the master regulator of cellular energy. When AMPK is activated, it goes and phosphorylates a very specific guardian enzyme called TET2.
To break that down, phosphorylation is basically the cell physically tagging a protein to change its function.
Yes.
It's like placing a chemical do not destroy sticky note on the TE2 enzyme. It tells the cell's internal waste disposal system to leave it alone.
That's a great visualization. Protecting TT2 is crucial because it is the enzyme responsible for active demethylation. It is the guardian that walks up and down your DNA, ensuring those 31,744 sentinel sites stay clear of blockades.
So the GLP-1 RAs shield the guardian, and the guardian protects the software.
Exactly.
So that is the broad systemic shield. But the paper also discusses highly targeted precision maintenance. They highlight a technology called Epigenetic.
Yes. Tiggertoss.
The best way to understand this is to think of CRISPR, but much, much smaller. Because it is so tiny and RNA-guided, you can attach it to specific enzymes and send it into the genome to do site-specific hardware resets.
Right.
It's like sending in a microscopic IT technician to fix exactly one line of corrupted code, say, on a highly critical gene like CD248 without touching a single other thing in the entire genome.
And achieving that level of microscopic precision is the holy grail of this field, because the blunt instruments we've used in the past are simply too dangerous.
Right, and the paper is very upfront about those dangers,
Right.
particularly when it comes to a class of drugs called histone deacetylase, or HDACs.
Right, HDAC inhibitors.
It notes that a drug called voranostat, which is a broad pan-HDAC inhibitor, can technically extend lifespan, but then it immediately warns that taking it carries a risk of fatal cardiac arrhythmias.
Yeah, significant.
It literally causes your heart to lose its rhythm. So if the goal is rejuvenation, why on earth are researchers experimenting with a pathway that can stop your heart?
Because the pathway itself controls how your DNA is physically structured, which is incredibly powerful. Histones are the physical spools that your DNA is wrapped around.
Okay.
If the DNA is wound too tightly, the cell can't read the software. If it's too loose, it reads things it shouldn't. An HDAC inhibitor relaxes those spools.
Oh, I see.
The problem with voranostat is that it's a sledgehammer. It indiscriminately alters how tightly the DNA is wound all over your entire genome.
Which sounds risky.
Very. You might successfully relapse an aging gene in your liver, but you accidentally alter the tension on a crucial ion channel in your heart tissue, and that's what causes the fatal arrhythmia.
So the goal is to stop using a sledgehammer and start using a highly specialized scalpel.
Exactly.
Yeah.
Broad pan-HDACs are a thing of the past. The future of this therapy relies on highly selective, class-specific inhibitors.
Like what?
Well, the paper highlights a novel molecule called CD60. Because it acts like a scalpel, targeting only very specific epigenetic locks, it doesn't just avoid cardiac toxicity, it actually rejuvenates heart tissue. Yeah. In systemic treatments, it reduced fibrosis in the kidneys and even reversed dementia-related gene expression in the brain.
That's amazing.
They also mention another molecule, Sanacrisma A, which is 3,600-fold more selective for its target than older drugs. It all comes down to absolute precision.
Okay, we've covered some incredibly futuristic high-tech interventions today. Epigenetic editing, microscopic IT technicians, blood plasma filtering.
It's a lot.
It is a lot, but I want to bring this right down to earth for you listening right now, because precision gene editing in a lab is amazing. But what is in your control today, in your own home, to protect your DNA methylation?
That's a great point. It is incredibly empowering to realize that your daily habits are already acting as natural epigenetic editors. Take fasting, for example.
Okay, fasting.
Intermittent fasting isn't just a mechanism for calorie restriction. When you give your digestion a break, you actively engage an epigenetic switch called the HIL-1H10 nutrient-regulated linker.
The HIL-1H10 switch.
Exactly. It is an evolutionarily ancient survival switch. By pausing the constant influx of fuel, you tell your cells to stop processing and start repairing. It optimizes the cellular recovery process.
So skipping breakfast might literally be giving your DNA time to defragment its hard drive.
That's exactly what it's doing. And then there's the microbiome. The paper points out that a healthy gut produces a short-chain fatty acid called butyrate.
Right?
And it turns out butyrate is a natural HDAC inhibitor.
Wait, really?
Yes, you don't necessarily need a synthetic, lab-created molecule to get some of those chromatin-relaxing benefits. You literally just need to feed your gut bacteria the right types of fiber, and they will manufacture the scalpel for you.
Wow, that's so cool.
Exactly. Your microbiome is a pharmacy. And for those looking at supplementation, the paper stresses the importance of protecting your NAD+ level.
NAD+, yeah.
NAD+ is vital for longevity proteins to do their jobs, but chronic DNA damage rapidly depletes it. So advanced protocols often include specific inhibitors to keep NAD+ levels high, along with compounds to protect your cellular maintenance enzymes from oxidative stress.
Right.
Even ensuring you have the right levels of vitamin B12 helps keep your biological software stable.
So what does this all mean? If we step back and synthesize this entire massive paper into one integrated protocol, we're looking at a deeply unified multi-stage process for systemic rejuvenation.
Absolutely.
First, you clear the toxic environmental sludge from the plasma. Second, you shield the cellular metabolism so the guardians can protect the software. Third, you use targeted therapies to rehabilitate the zombie cells. And finally, you clear the metabolic waste so your immune system can let those telomere rivers flow, irrigating your aging tissues with localized youth factors.
It is a profound, actionable roadmap for healthspan extension. But I want to leave you with one final, truly fascinating detail from the sources that ties everything we've talked about together, from the complex biochemistry right down to how you feel when you wake up.
Oh, I'm intrigued.
We just mentioned that your gut bacteria produce butyrate, that natural epigenetic editor. What researchers found is that this butyrate production isn't constant. It has circadian fluctuations.
Really? It operates on a timer?
Yes. It rises and falls in rhythm with your biological clock. And incredibly, the peaks and valleys of that bacterial butyrate production negatively correlate with the severity of morning stiffness in human joints.
Wait.
Yeah. The lower the butyrate, the stiffer the joints.
That is wild.
It implies something massive for the entire future of chronobiology. It suggests that the time of day you apply a rejuvenation therapy, whether it's a drug, a supplement, or a fasting window, might be just as important as the therapy itself.
That is incredible to think about. The future of anti-aging, the key to unlocking that corrupted software and turning back the biological clock, might not just be about finding the perfect molecule.
Yep.
It might be as simple and as elegantly complicated as setting an alarm clock for your microbiome.
Exactly.
Well, thank you so much for joining us on this deep dive. To you listening, keep questioning the paradigms, keep learning, and stay curious.